Hello, my name is Dr. Jim McClintock. I'm the professor of polar and marine biology at the University of Alabama at Birmingham, and I'm excited to share with you a little bit of the story of how I got interested in science and how my interest in science eventually led to a career working in one of the most fascinating places on our planet, Antarctica. Before I talk about Antarctica, though, I want to tell you a little bit about my years growing up as a kid and how this might have influenced my interest in science. So I raised in Santa Barbara, California, which is on the coast and surrounded by mountains. There was a wonderful creek that ran through my neighborhood. And I think being surrounded by such beauty and nature was part of the reason that I took an early interest in nature. I was always that kid that went down into the creek and caught frogs and turtles. I remember I even dug a little turtle pond in the backyard and kept frogs and turtles in it for observation, for fun. And I also had an interest in things like insects early on. And I'd go through the fields looking for bugs, looking for beetles, dragonflies. I can remember I had a net that I would carry to capture insects and look at them. And the creepy crawler ones were particularly exciting, things like the worms that you'd find. I don't think there's a rock around my house in Santa Barbara that I didn't turn over at some point trying to find the animals that lived under it. And this curiosity in animals started very young, and I was asking questions like, well, why are they under the rock? What is it about that they like? What are they eating? How do they behave if you put them together? What do they do with each other? So I always thought it was fascinating to see exactly how these animals made their living. I also was interested in the ocean very young, and I can tell you that in Santa Barbara, there was plenty of ocean to explore along the coastal wet sands. I would chase mole crabs that were digging into the sand as each wave disappeared. And this is actually a picture taken of a blue crab here along the coast of Alabama, one of the species of crabs that are very common here. And I loved finding shells and seeing who was living in them. Sometimes the animal that made the shell was not the animal that lived inside it. I'm holding here a moon snail shell, and the animal living in it is actually a hermit crab. So fascination with these organisms that were living along the coast. A very formative part of my early life also was scouting. And after being a Cub Scout for several years, I joined the Boy Scouts, and it brought two things to bear. One was that the merit badge system was a great way for me to learn early rules about science, because I was collecting information, I was putting it into some sort of order, I was writing it up and finishing the steps of reaching a given requirement for a merit badge. And this is very much like doing science. And so that was a great way to learn about some of the rules that are involved in guiding scientific inquiry. The other thing about the Scouts that was very formative for me was camping. And this took me out into the nature. It could be as simple as an overnight camping trip just outside of the neighborhood, to a 5-day backpacking trip in the high Sierra Mountains, hiking maybe 50 miles over a period of a few days. What an amazing experience to see the wildlife, to do some fishing together, and to experience nature of this magnitude. So that was very formative early on with scouting. I also discovered fishing when I was probably 5 years old, and my dad would take me fishing in the mountains. And I also fished in streams and lakes that surrounded our house. There was the little creek nearby. There was a pond up at a neighboring golf course where I would sneak on and hide in the bushes as I fished for bass. So fishing taught me a lot about nature. I was interested not only in the fish that I caught, but what they were feeding on. So I learned about the different kinds of foods that the fish consume. And today I still fish. It's very much catch and release, where you catch the fish and you let it go. It's more of a sport. And I'm still learning things about natural history and about how fish make their living from different fishing experiences. It's also been a wonderful way to share time with family, such as this picture of me in New Zealand, where we lived while I was doing a research sabbatical at the University of Otago. And here's my son Luke to my right, helping me hold this big rainbow trout that we've managed to land in this reservoir in New Zealand. So fishing, very important. Another stage in my growth as a young scientist was when I got to college. And here I took the opportunity to go up the coast and spend an entire semester, 3 months, living and working at a marine biology station. There were 20 students from the University of California there with me, and we were completely immersed in marine biology for this period of time. We had wonderful teachers that taught us about marine biology, and even more important, we were allowed to do our own research projects. And I can remember mine was working in the rocky tide pools. I was looking at the growth of sea urchins in different tide pools and trying to understand why they grew at different rates, depending on where they were in this rocky intertidal. Most important, we were learning how to do science, how to formulate an important question, how to address that question, how to set up an experiment, how to record the information, and more importantly, how to interpret that information in the context of the environment that the animal lived in. This was how science worked, and I was excited about it. So it wasn't too surprising that after being here and spending time in the rocky intertidal and getting addicted to marine invertebrates, those animals that don't have backbones, the 95% of the animals that are out there, that I went on to the University of South Florida to do my doctoral degree in marine biology, and then did a postdoctoral fellowship back at the University of California at Santa Cruz, where I had been an undergraduate. And by the time I looked for positions as a professor, I had been to Antarctica 3 times, had published many papers on the topics, and I was in a great position to go on from my marine biological early years to become a professor of marine biology. This was facilitated, of course, also by an early interest in reading. And one of the most influential authors in my early life was Professor E.O. Wilson, who was at Harvard, but also is connected to the state of Alabama, where I live now, where he was born and grew up and went to college. Many of his books are outstanding books for young people to learn about natural history and about the excitement of doing ecology. Here's my picture of my class that I teach in the Galapagos. I'm in the back row there, and my students are reading The Future of Life, an E.O. Wilson book. I'd also recommend that you read his book called The Naturalist, a wonderful introduction for young people who are thinking about going on into science. And E.O. Wilson is the ultimate ambassador when it comes to learning science and its importance. So here's a picture of my university, where I have spent my career now for 35 years, the University of Alabama at Birmingham. And the university has been very important to me as a place where I could do my research in the laboratory and do the courses that I love to teach, and also to serve as a transit point from Antarctica, where I did much of my research over the years. So let's think then about Antarctica. Let's lay the foundation of what this place is. Antarctica, unlike the Arctic, is a continent surrounded by oceans, whereas the Arctic is an ocean surrounded by continents. So it's very different. The continent of Antarctica, at the bottom of the Earth, is massive. It is the size of India and China combined, if you can imagine. It's otherworldly. It's very much like visiting another planet. It's an amazing place to do science because so much of the science there is new and so many discoveries have yet to be made. Well, let me tell you a little bit about the places in Antarctica that I've worked. My first trip to Antarctica was actually to the subantarctic. And if you look at the arrow up in the top right-hand corner that points to Africa, Just to the left of the tip of the arrow, there's some islands, and one of those islands is Kerguelen. And Kerguelen is a little subantarctic island that I visited when I was a graduate student. And I spent 3 months living and working at a French Antarctic station with my major professor, John Lawrence, and we studied all sorts of things from the intertidal ecology to the reproductive biology of different kinds of echinoderms, starfish, and things like this. And it was a really great place for me to get excited about doing work in Antarctica. I then moved my research to McMurdo Station, which is down at the bottom of this picture. McMurdo Station is the largest of the 3 American stations. There's 1,000 Americans there during the summer. It's also surrounded by sea ice and very, very cold. In order to get into the ocean at McMurdo, you really have to drill through the ice in order to get into the ocean. The last 20 years, I've been working at Palmer Station. So look at the map and look way up in the left-hand corner, and you'll find Palmer Station. It's on that 800-mile long Antarctic peninsula below South America. And it's a warmer region of Antarctica, and a region of Antarctica that's changing very rapidly with climate change. And I'll tell you a little bit more about that as we go along here. So Antarctica is a world with ice. And I must remind you that the entire continent of Antarctica is covered with a sheet of 2 to 3 mile thick ice. It's so heavy that it actually sinks the continent. Now, there's all sorts of different kinds of ice. The ice that sits on top of Antarctica are known as ice sheets. And then along the coast, you can have very large glaciers that extend to the sea. This is a picture of the Mar Glacier, which is just behind Palmer Station where I work. And the Mar Glacier has been receding very quickly because of the warming that's going on in this part of the world. In fact, about 90% of the glaciers on the Antarctic Peninsula on the west side are now receding. That means that big chunks of ice are falling into the ocean, and the glaciers are pulling back as it warms. In addition to the glaciers, there's sea ice. Now, sea ice forms very shallow. In other words, it's only about 5 to 7 feet deep, and it doubles the size of Antarctica in the winter. It forms on top of the ocean and doubles Antarctica's size every winter. Imagine that. And then it breaks up and it goes out to sea in the summer. So it's a very dependable annual process, and many of the marine organisms in Antarctica are dependent upon the ebb and flow of the sea ice for their lives. And I'll mention one in just a couple of minutes for you. So lots of things are built around the ice. In order to get to Palmer Station, unlike the planes that we flew on to get to McMurdo Station, we use ships. And the National Science Foundation provides 2 ships that we use to cross the Drake Passage. We start in Chile, in a little town called Punta Arenas. We board our ship, and it takes us about 4 days at sea in order to get across the Drake Passage. It's about a 700 to 800 mile long passage of some of the roughest oceans in the world. One winter, I came back in 45-foot seas, if you can imagine that. I couldn't even stay in my bed because it was so rough. So once we get across the Drake Passage, and normally it's not too bad, we arrive at Palmer Station, which is our home away from home. And there I'm usually met with my 2 colleagues that make my science in Antarctica such a wonderful task. My work in Antarctica is done as a collaborative team, and the fellow in the middle of this picture's name is Bill Baker, is a chemist, and the fellow on the far left is Chuck Amsler, and he is a plant or seaweed biologist. They're called phycologists. So the 3 of us, myself on the right, Bill Baker in the middle, the chemist, and Chuck Amsler on the left, the seaweed biologist, have worked together for 25 years to try to solve the riddles of Antarctic marine chemical ecology. Why do marine animals and plants or seaweeds produce chemicals that are toxic? So this has really been the focus of our work for many, many years. Now, to get into the ocean, to get the animals that we study, we have to wear a very special suit called a dry suit. And the dry suit Chuck is putting on in this picture here has a very tight neck seal on it to keep you dry. You're wearing underwear underneath the rubber suit. And on the chest, you can see a place where the hose from the tank will hook up. And the way that you stay warm in your dry suit is you pump it full of air. And your warm underwear keeps you warm underneath that layer of air. Now, because you have air in your suit, you are very buoyant. You're a cork. So you have to wear about 30 to 40 pounds of weight around your waist, and you also have to wear weights on your ankles in order to get down below the surface of the water. Here's a picture of us preparing to go for a dive a long time ago. This is an outdoor dive in McMurdo Sound. I'm sitting on the left, getting my tank on and getting ready to slide into the hole and into the water that's, about 30 degrees Fahrenheit. That's right, it's sub-freezing, it's below freezing water. And the reason that the water can be sub-freezing and not frozen solid in an ocean is that there's salt in the water that lowers that freezing point. Now, if we're diving at McMurdo, we have to first dig a hole to get through the ice. And if we're near the station, we can do this with a big drilling rig that will drill about a 3-foot diameter hole. And then we can take a dive hut and put it on top of that hole. And we can go into our little warm dive hut, has a heater. We can get into our dry suits, drop through the floor of the hut and down about 100 feet to the bottom of the ocean and collect our animals and then return to the hut. So it works quite well when we're near the station where the big drilling rig can go. Now, when we go further out into the field to collect animals that don't occur near the station, we go camping. And we have often used a helicopter to take us out into the field and drop us off with all of our dive gear and our camping gear. And we set up a campsite for 4 or 5 days. The first thing that we have to do is to get through the ice for our diving. So we take along a little drill called the Jiffy Drill, and it's got about a 6-inch diameter bit. And once that hole is in the ice, we take about 100 pounds of dynamite and we put it down into the hole, and then we step back and we explode the hole. Fortunately, all that energy goes up, and we don't do any sort of work like this near seals or penguins, so we don't hurt anything. And then when you come back to the hole where you've done the explosion, you have to work very hard to clear all the chunks of ice and snow away and see if the hole's big enough for scuba divers to fit through. If it's not big enough, you have to use dynamite again. So you get the hole established, and then you can camp near it, and you can spend 3 or 4 days collecting your animals and seaweeds, and then take them by helicopter back to the lab where we study them. And here's a picture of one of our field sites that was in the background here. You can see the big tent on the right where it was our kitchen and sleeping quarters for some of the members. Others were sleeping in little pup tents off to the left. And in the middle there, if you look closely, you can see a Scott tent set up, which also is used as part of our campground. Here's a closer image of the Scott tent, named for Robert Scott, who tried to be one of the first people to reach the South Pole and tragically lost his life in 1911 or early 1912, and on his return from the South Pole because of a very, very bad storm he and his men got caught in. So we were camping in order to collect. This is a picture of our field team from one of the many years we went down to McMurdo. You can see it's a group of folks that includes students and postdocs and research professors, divers, maybe even a carpenter. They're all essential in order to make such a field-intensive project work. We also had access for a 2-year period to this hovercraft. And here you can see a picture of the divers and myself on the left preparing to get in the hovercraft to go for a scuba dive. It was a wonderful way to get to a dive site and be able to get dressed inside a vehicle that was warm. That was a great thing to have as an asset while we were down at McMurdo. Once you're diving under the water, the visibility is absolutely stunning. You can see 100 to 1,000 feet underwater, depending on the conditions. It makes it for the clearest water in the world in order to dive. You can see that a safety line has been dropped from the hole. The safety line is equipped with flags, and on the bottom there's a strobe light flashing, because you certainly don't want to lose sight of the hole while you're scuba diving along the seafloor in order to collect the animals that we're studying. Here's a picture that really gives you a sense of the visibility underwater. A diver that is silhouetted against the ice above, and you can see the hole that they came down through. Just an absolutely stunning place to dive. Now, once you're on the seafloor, there's some things that are quickly obvious, and one of them is that down to about a depth of 100 feet, the floor of the ocean is covered with what we call anchor ice. And that anchor ice really keeps any of the life from existing in that zone. It's something to do with the depth and the temperature combination that creates ice platelets that form on hard objects. So you can see here that the sea anemones in this picture are in deeper water, and the anchor ice is in shallower water. And here on the left, you can see some more of that anchor ice creeping down towards the seafloor. The sea urchins in front of it are just out of the reach of that anchor ice. On the seafloor, there is a myriad of sponges and soft corals and starfish and sea urchins. One of the first things that I thought of when I dove was that this is amazing in its diversity. The diversity of life on the seafloor of Antarctica is unparalleled. In many cases, the amount of sea life is comparable to the Great Barrier Reefs of Australia. Another wonderful picture of the anchor ice that's shallow, and one more showing you the beautiful visibility as you go up against the shoreline. And now we're going to take a look at the animals and the seaweeds that occur on the bottom. So just pretend that you've put on your scuba gear and you're accompanying me on a dive underneath the sea ice. So this is a giant brown seaweed that has blades that are about 10 feet long. A lot of people don't realize that there are underwater forests in Antarctica. In fact, along the Antarctic Peninsula, there are about 150 species of seaweeds, browns and reds and greens. And this is another picture of a brown seaweed that grows here. Notice these little balls on it. They're called nematocysts. They're used as floats to keep the seaweed up in the water. There are also red seaweeds. This is one of my favorites. It's a coralline algae, a coralline seaweed. And it's a red alga that makes a calcium carbonate, a rock-like material, and glues itself down to the substrate. And here you can see a close-up picture of that with some fleshy algae as well. Aren't those bizarre-looking animals, or plants, I should say? Look at these crazy-looking red coralline algae. Speaking of animals, wow, the sponges of Antarctica are amazing. One of the most biodiverse groups in the Antarctic seas. Often the seafloor is covered with a mat of sponges. The sponges are all different colors, yellows and reds and oranges and tans. There are many different forms. An unusual thing about Antarctica is that sponges that occur in the deep sea can also be found in shallow water. So you can find things like this giant glass sponge. This vase sponge is normally something that you'd see by submarine very deep, but here it is taken, a picture taken in shallow water. These vase sponges can get so big that a scuba diver can fit inside the vase, 5, 6 feet tall, 4 feet across, growing very slowly, which would indicate that they are very old animals. They may in fact be the redwood trees of the deep Antarctic waters. Some of the sponges are protected by spines, or called spicules. And here's an example of one that's protecting itself from predators using these sharp spicules. Most of the sponges in Antarctica don't use spicules to protect themselves, but they use defensive chemistry, chemicals that taste bad to predators. For example, sea stars, which might like to eat the sponges. And some of the sponges are in fact eaten by these sea stars. Sea stars have an interesting way of feeding. They take their stomach and they push it out of their mouth against the food that they're eating. Now, aren't you glad that we don't do that? That would be kind of messy, wouldn't it? But these sea stars are very good sponge eaters, and it's often that you see the sea stars perched on sponges. Some of the sponges have some very smart ways to avoid the sea stars. For example, this circular sponge that's orange is the top of the lollipop sponge. It grows on a stalk, and the stalk is chemically defended to keep the sea stars from climbing up the stalk and eating the sponge. So it's a wonderful way to avoid predators. Many of the sponges in Antarctica also have the potential to have chemicals in them that could be drugs that fight human diseases. And here's one example that we have discovered. This is the cactus sponge, Dendrilla, and it produces a chemical that's very, very active against a bacteria called MRSA. And this is a problem because MRSA is the type of bacteria that's hard to treat with antibiotics. So this is exciting to see that there's antibacterial compounds in Antarctic sponges that could become drugs to help fight against infection. And among the other organisms besides sponges that don't move around much are the Cnidarians. And here's a beautiful picture of a sea anemone on the seafloor of Antarctica. These sticky tentacles are used to capture prey, and I've seen them catch sea urchins. And in one case, I watched them catch a jellyfish and eat it. This sea anemone, you can see the eggs that are produced by the sea anemone, and they'll be released into the water to be fertilized and to grow into young sea anemones. There are soft corals that belong to the Cnidarian group. There's no hard corals in Antarctica like you'd find on a coral reef because there's not enough sunlight to support those corals and the little photosynthetic plants that live in their tissues. But there are soft corals like this beautiful one. If you look up in the water in Antarctica, you might also see things like a comb jelly. This beautiful little jellyfish-like animal has little rows of cilia that stream down its sides that are iridescent, greens and blues and yellows, and they beat very quickly, and they are what you use for propulsion for moving around. These comb jellies can be very common. Another very common animal that swims around in the water in Antarctica is the sea butterfly. And here's a beautiful little sea butterfly that has no shell. It's a naked sea butterfly, and it doesn't taste good. It's got a chemical in it that fish do not like to eat. So you can imagine the surprise when a friend of mine, John Jansen, and I discovered a little amphipod, a little crustacean that you can see in this picture that captures and carries a live sea butterfly. So you can see that it's got its legs attached to the sea butterfly in this picture. And if you remove the sea butterfly and you offer the little shrimp-like amphipod to a fish, it'll eat it every time. But if you leave the sea butterfly on the back of the little shrimp-like animal and you offer it to a fish, the fish will take it in the mouth, taste that nasty sea butterfly, and spit them both out. So it's a very smart little shrimp-like animal. It's figured out how to abduct and carry around another species on its back for its own protection. Wow, this is so unusual. Antarctica has many surprises. Antarctica also has sea slugs that are chemically defended. Sea slugs don't taste good from all of the world's oceans, and Antarctica is no exception. This one was so funny to me. It reminded me of sort of Uncle Harvey, a picture of a human face, but actually a sea slug in Antarctica. There are some unusual things in Antarctica that get very big. One of them is this Antarctic worm called a Mcmurdo. This worm in Antarctica gets up to 10 feet long. This is an example of what we call Antarctic Gigantism. This worm has a nasty mucus that has a very, very acidic nature to it. So nobody eats this worm. And this worm is very much a predator on other animals on the seafloor. This is an Antarctic polychaete. It's called a feather worm. And those feathery things on the top are used to capture food. And this is an Antarctic isopod that I think looks a lot like a trilobite, and a group of organisms that used to be very common in the world's oceans but have gone extinct. This is an Antarctic isopod crustacean. Another crustacean in the Antarctic seas are the isopods that are very common. Here's a little isopod perched on a soft coral. And of course, the amphipods everywhere. These are important in decomposition of marine life. In other words, when things die, amphipods tend to eat them and break them down and turn over the nutrients and energy in this system. So amphipods are very, very common. Maybe the most important organism in the entire Southern Ocean is the Antarctic krill. This is called Euphausia superba. It kind of looks like a shrimp, but it's not a shrimp. They are so common that they form massive, massive swarms in the ocean, the size of city blocks. These little animals, about an inch long each, form the base of the food webs in Antarctica. Everybody eats krill. Whales eat krill. Seals eat krill. Penguins eat krill. Fish eat krill. Starfish eat krill. It goes on and on and on. So they're very, very important to providing nutrition for the many different food levels of Antarctica. We have discovered that when krill are teenagers, they live under the sea ice. You can see a group of teenage krill underneath the ice here feeding on little diatom plants. Now, this is important because as climate is warming here in Antarctica, the sea ice is disappearing in some places. And this means that there's not habitat or places for these juvenile krill to live, and it could have an impact on krill in the future. This is an Antarctic sea spider at the base of the picture. These can get up to the size of a dinner plate in Antarctica, another example of Antarctic gigantism. My favorite group of animals in the ocean are the Echinoderms, which includes sea stars. Look at this beautiful rose-colored sea star. It's a very successful species called Odontaster. It's successful probably because it has many ways of making a living and eating. Here you can see its arm tips held up in the water, filter feeding out little particles of food. It can climb onto sponges and eat them. It can eat dead and decaying organisms. It's a fantastic, opportunistic feeder, and this is probably why it's so common. Here you can see an Odontaster perched on a sponge. If I was to turn this over, probably its stomach is extruded and it's digesting that sponge. They also interact with one another. These sea stars actually fence with their arms. They fight with one another to see who wins the battle, and the winner of the battle gets the food that they're fighting over. Who would have guessed it? And some of the sea stars have more than 5 arms in Antarctica. Look at this beautiful Antarctic sun star with many, many arms. It can hover over the seafloor. It kind of pulls its arms in and forms a little cave under itself. And when a fish comes into this little space, the entire sea star drops onto the fish and has lunch. And here's an Antarctic sea urchin. Look at those big spines it uses to protect itself. And Antarctic sea cucumbers, like sea cucumbers everywhere, have the most unusual behavior. It's called evisceration, where they can spit out their entire internal guts. Their stomach, their ovaries, the intestines, everything comes out when they're disturbed by a predator. So the theory is that they spit out everything to offer up something good to eat to the predator, and then they slip away and they can regrow all their internal anatomy. It's absolutely amazing in order to continue to succeed and be a successful organism in that habitat. The crinoids, the feather stars, are also abundant in Antarctic waters. And this species, when I swim over with my scuba gear and touch these arms, they begin to wave, and pretty soon, like a helicopter, the entire feather star lifted off the sponge it was sitting on and swam about 10 feet away. This is what we call an escape response, running from a predator. Now, the sea squirts are one of the final groups I want to mention. And these animals, known as tunicates, have also been the source of a chemical we described as palmarolide. Palmarolide has activity against cancer, and particularly melanoma skin cancer. So it's another example of chemistry in these Antarctic marine organisms that could help humanity in the future, and something, it's a resource that we need to protect. Fish, of course, occur in Antarctic waters. There's 250 species of fish. If you think about it, fish should have a tough time living here, and in a sense they do. The water is below freezing, and so their blood should be frozen solid. But a scientist in the 1960s discovered that they have an antifreeze in their blood that keeps the blood from freezing, just like you'd put in the radiator of a car at cold temperatures. So this has allowed fish in Antarctica to exist. There's some very unusual fish in Antarctica, including what we call the ice fish. And here you can see an ice fish. The ice fish are clear because they don't have hemoglobin. They don't have red blood cells in their blood that carry oxygen. This is thought to be because the water is so cold and so full of oxygen that they just simply didn't need to have red blood cells, and over time lost them. So it's quite an unusual physiology. Of course, penguins, everybody thinks of in Antarctica, and there's 4 different penguins I want to introduce you to. These are the emperors, the largest of the penguins. They stand about 3.5 feet tall, beautiful, majestic birds. They live very far south. They are truly an Antarctic species, feeding largely on krill and fish. And then there are 3 species of what we call brushtail penguins. And the 3 that I want to introduce you to all occur around Palmer Station where I work. This is my favorite, the Adelie penguin. It's got a little black and white feathered arrangement that makes it look like it's wearing a tuxedo. And look at the rock. The rock is there because males offer females rocks as part of a mating ritual. This is the Gentoo, a little bit bigger than the Adelie penguin. This is a subantarctic species. And the 3rd is the chinstrap penguin, again, about the size of the Gentoo penguin, about 2 feet tall, and again, on the Antarctic Peninsula. I want to tell you a quick story about what's going on with these 3 brushtail penguins near the station where I work. The one up in the top left-hand corner, the picture of the Adelie penguin, is depicted in the purple line on the graph. And there's been a very long-term study that has followed the numbers of these penguins. And you can see that the Adelie penguin in purple has been disappearing, almost to the point now where we're down to about 1,000 pairs in 2021 from the original 16,000 breeding pairs. About 90% of them are gone. At the same time, the penguin in the far right picture, the chinstrap in the green line, has increased in number. And the penguin in the middle picture, the Gentoo, has really increased quite a bit. And there's now over 1,000 breeding pairs near Palmer. So what's going on? Well, the picture of the Gentoo and the chinstrap exemplify 2 penguins that are warm weather penguins that are moving in as climate change is warming the region. And the Adelie penguin in the top left-hand corner is a colder weather penguin that's having a tough time as it warms. And the reason that the Adelie is having a tough time is largely because the sea ice is disappearing, and they use that sea ice to scoot on their bellies out to the edge of the ice where they feed. And they're also getting covered with snow, and it's burying their eggs, which is making it difficult. As it's warming, it's more humid and it's snowing more. So good news for the other 2 species of penguins, but not such good news for the Adelie. When we're diving around Palmer Station or at McMurdo, we're always aware of leopard seals, 10 feet long, 1,000-pound top predators. If we see a leopard seal, the first thing we do is we set off a siren to warn the divers it's time to come up. And they get together back to back and wait for the leopard seal to go away, and then zoom, up they come and into our Zodiac boat, and we move to another place. Because leopard seals can be very dangerous and territorial, but they're beautiful animals. And of course, we can't forget the whales of Antarctica, such as this beautiful humpback whale. The story of whales in Antarctica is a good story at the moment. The whales were hunted almost to extinction back around the late 1800s, early 1900s. And whale biologists in Antarctica believe that time has now allowed the populations to begin to really recover. And I have to admit, over the last 5 or 10 years, I have seen more and more and more whales when I go to Antarctica. I counted over 60 humpback whales in a single afternoon one day. Beautiful whales that were spouting, jumping out of the water or breaching and feeding on krill by forming bubbles that form cylinders, and then that makes the krill scared. They come together in a ball, and then the whales just lunge up through the ball of krill, feeding on them. It's spectacular. So what an amazing place to have studied marine biology, Antarctica, a real treasure and really a place where we have very much yet to learn. I want to end my talk with you with just a quick story that makes you think about keeping the fragility of our planet in perspective. A year and a half ago, I won an award on my Antarctic research and my educational work in Antarctic science that was awarded to me by the Explorers Club. And I went up to New York City to receive the award in front of an audience of about 1,500 people. And sharing the stage with me that day were 8 of the living Apollo astronauts. And what an honor it was to get to meet them and talk to them. And one of the things that they did at the end of the evening was to have each of the astronauts tell a story that nobody may have heard before about an adventure they had had. But when we got down to the last astronaut, this fellow right here, his name was Rusty Schweikert. He's got the microphone. And Rusty became very serious. And he let the audience stop talking. The room became silent. And he stood up and he looked at his fellow astronauts and he said to them, you know, all of us here on the stage have been up into space, and all of us have looked back on this planet, and we have seen with our own eyes its fragile nature. In other words, it's very susceptible to what we do to it. There's a very tiny, thin biofilm, or what we call the biosphere, almost looks like a biofilm, the biosphere that surrounds the planet, that supports life on it. And so we must work together, all of us, to keep our Earth happy, intact, and healthy. And I'll end on that note. So good luck. Hope you've learned something about Antarctica. Goodbye.